Hello. The presentation is about SoftMIL, the algorithm for predicting time to biochemical recurrence of the prostate cancer based on whole slide images of the prostate tissue. The solution was developed as a part of the Leopard Challenge competition. Before we jump to the solution, I would like to say a few words about us, about me. My name is Kamil Rzechowski and I'm Senior Machine Learning Engineer in SoftwareMill. And in Reason Field Lab department, we do various AI solutions. For example, we had the projects in medical AI domain. We did tumor cell segmentation of whole slide images. We did 3D astrocyte segmentation. Right now the challenge is about predicting time to cancer biochemical recurrence. We had the project about predicting cancer metastasis. And all of those solutions are developed using explainable AI. So we explain based on which assumptions the model did the prediction and verify whether those assumptions are correct. And so using Psi also helps us during the development process to boost the model performance. Jumping to the other side of the screen, we have generative AI, very famous right now, retrieval augmented generation chatbots and agents. We do customer tailored custom solutions. We can do self-hosted chatbots. We can implement those using the cloud solutions. We have also experience in generating images using stable diffusion. Here an example of interior design. Given a sample image, we can generate multiple versions of the same room in different design scenarios. And of course, we do as well classical machine learning, XGBoost, random forest, decision trees, also time series processing and predictive algorithms, and we do productization, so automation of ML workflows, continuous integration, continuous deployment, continuous training. So the competition Leopard Challenge was about predicting time to cancer biochemical recurrence based on prostate tissue whole slide images. And the evaluation was done using concordance index. In simple words, concordance index is the measure of how well patients in this case are sorted based on the time to biochemical recurrence, and in case no biochemical occurrence appeared, those patients are at the end of the sorted list. Here is an overview of the solution. So this is the multi-instance learning framework. As an input, we have the whole slide image tissue of the prostate. And the whole slide image is divided into multiple patches of size 224 by 224. Then we use the frozen feature extractor. In this case, it's PLIP. The clip, the pathology version of the clip model. And those frozen features are then fed to attention layer, trainable attention layer. The attention layer picks up patches that carry the information required to make the prediction. And the output of the attention layer is fed to classification layer. The classification layer, trainable classification layer, predicts whether there will be or no the biochemical recurrence. And then finally, on top of that, we have the regression layer that predicts time to biochemical recurrence. Because For the computation of the concordance index, we use the classification layer. It worked really well. First of all, because we had a really good correlation between, actually inverse correlation between output of the classification layer and time to the biochemical recurrence. And it also worked really well because the dataset was highly disbalanced. Only about 10% of cases had biochemical recurrence. So the regression layer, the information to propagate back the regression layer was very sparse. However, for the classification layer, we had all the samples, so the information was like, let's say, the whole. We possessed the whole information. But the regression layer was necessary and improved the classification layer results significantly, and improved also the inverse correlation between classification output between 0 and 1 and the time to biochemical recurrence. About the training strategy, we used the hard samples masking strategy. This was taken from the paper Hard Samples Masking, Multi-Instance Learning, something like that. The hard samples masking is a popular technique in computer vision. It is used to force the model to learn based on hard samples, no easy to classify samples. It works in our solution. It worked in the way that we sorted all patches based on the attention score, from the highest attention to the lowest attention, and then we masked some of the high attention patches, and therefore force the model to make the predictions on the samples that had lower attention, so basically were less useful, carried less information for classification purposes. And on the right we can see the sign scheduler for hard samples masking ratio. At the beginning of training, the attention scores are pretty random, so it doesn't make sense to mask any of them. The model doesn't know which to use anyway. And over the training, we increased the ratio of high attention masks that were filtered out. And therefore, we forced the model to learn a better discrimination meaning fault. Another technique that was used during the training process was firstly the stratified cross-validation. We divided the dataset into 3 folds, each fold containing training, validation, and test set. And during inference, we've taken 6 different models, 2 per each fold, and based on the validation dataset, and we did inference. We propagated the inputs through each of those models, and then those models voted for the output. And I had some issues with internet connection and slides, so sorry about that. The architecture of the model, the attention layer inside the architecture, provides the explainability of the model by design. And here it's actually very interesting because from the whole slide image, only a small part of the tissue highly contribute to the result. Here we can see like the main spot of contribution, and here there is like 1, 2, and 3 spots with that also contributed to the final prediction for this tissue particularly. It would be also very interesting to verify whether those attention maps correlate with tumor cells. However, I didn't have the ground truth for tumor cells. Neither I had algorithm for predicting tumor cells. So this is like left for as an open question whether those attention maps correlate with tumor cells. Hopefully they do. And about results, this is the validation. Those are the results based on the Leopard challenge validation set provided. The validation set was not used for hyperparameters tuning, neither for picking the best model or anything like that. So this is like completely separate test dataset. The concordance index, the average concordance index was 0.62. The internal, the same cohort dataset that the model was trained on, the C index is 0.64, and the external cohort, so external dataset, the concordance was 0.60, so we can see here a slight distribution drift. Thank you very much for listening my presentation. I hope it was useful. In case any questions, I'm very happy to discuss it. You can contact me personally or through hello@softwaremill.com, or in case of any other medical AI problem, also feel free to reach out to me. Thank you very much.